Hi, good morning or good afternoon, wherever and whenever you are watching this presentation. I appreciate your interest in what I'm going to present for you today. I'm very excited about the work and the outcome of the work that stems from a new de novo genome assembly of NC94022. And it's not just about a genome, because we have genomes, but it's about the power of de novo genome assembly for peanut in terms of identifying functional variation for important traits, and in this case, TSWV resistance. And really the leader of this work is Baoju Guo, who's been spearheading mapping this resistance for a while now and really providing the leadership to get this done. And his group has been essential in the work, as well as Brian Scheffler's group, the USDA ARS GBRU, Cal Youngblood, and Sharon Simpson. Albert Colbreth, eminent pathologist, University of Georgia, and of course Juliet Chu and Peggy have contributed as well. So those of you that don't know yet, I'm officially sort of announcing my new position as faculty at Hudson Alpha Institute for Biotechnology in Huntsville, Alabama. Those of you that aren't familiar with Hudson Alpha, it's a nonprofit institution that mostly is focused on human genetics and genomic health. And it's also the site of Jeremy Schmutz and Jane Grimwood and their team that has been sequencing plant genomes for over a decade now, and really a leader in that space, and the ones that really unlocked the ability to have the Tifrunner genome finished, and they were the ones crucial to doing that work. So I'm excited about being here and the potential for the research in terms of what we'll be able to do at Hudson Alpha with their sequencing capacity, with the brain power, and with the resources they're able to leverage. And I'll still be focusing in on using genomics to improve crops, focusing on peanut at least for now, but expanding to other crops as well. For the topic at hand, NC94022 is a hirsuta derived breeding line that is extremely resistant to TSWV. I'm showing work from 2005 with locations in Florida as well as Georgia, showing just how strong resistance NC94022 has. And then there's a picture on the right there of NC94022 and Senoleg 97R, really showcasing how brilliantly it resists TSWV. There's been quite a bit of effort to map this resistance at Florida and in Tifton by Baoju Guo. There's many papers that have done a fine job mapping QTL, all on chromosome A1, very strong. So this is a major gene that resides on A1. It's very clear. I'm not going to show all those papers, but just this one here from Baoju and Gaurav Agrawal, where they used whole genome resequencing of Baoju's T population, which is a cross between NC94022 and Senoleg 97R. And they were able to really nail down the QTL. The region of the QTL is about a 250 kilobase region. Again, the resistance to TSWV has been confirmed over and over again, recently again in 2019, looking at some populations where the parent, the CAPS populations that led by Corley and Tom Islip, but the parent is actually SSD6, which is derived from a PI, which was the initial parent that was used to generate NC94022 and contribute that resistance. And so this is another nice picture showing just how resistant to TSWV NC94022 is. And so what was really intriguing about these data is this particular test that they did last year in Tifton. They had a bunch of interspecific lines, some from Tom Stocker that had Diagoy integressions that Tom has been very excited about with the TSWV resistance. Some other sort of interspecific amphidiploids, and then the real population from the CAPS rails. And what they found is, of course, NC94022 is the best, and it even performed better than the Diagoy interspecific lines, which is really pretty unique in the sense that the interspecific lines for these types of resistances are usually much stronger than what's found in the cultivated germplasm. So this is that rare occurrence where actually it appears as if the cultivated resistance is the strongest. And that holds up also in the real population with SSD6. So again, the resistance is incredibly strong. It's been tested over and over and over again in multiple locations in Georgia and Florida, in multiple sort of genetic backgrounds. And so really excited about the potential of having functional variation to use marker-assisted selection to incorporate this into more cultivars. So last year, Baoju engaged me about fine mapping. I was a co-author on the Agrawal paper in which I helped them essentially analyze the SNP data and get the genetic maps and really do the mapping with the short read sequencing. And Baoju really wanted to fine map that and get a gene. And I said, I think the only way for us to do that is to generate a parental genome sequence de novo. And so he, as he is wont to do, a very future, so far-seeing researcher, he said, yes, let's do it. And so Brian Scheffler sequenced NC94022. We assembled it into a genome about the size that we expected, with N50 close to 2 megabases, and even an N90 about almost 600 kb. And basically what that means is that 90% of the genome is in contigs that are larger than 590 kb. So very contiguous genome sequence. We're very happy with the results. And as you can see, we scaffolded with Tifrunner, sort of a reference-based scaffolding. And I'm just showing dot plots with chromosome A1 and chromosome A2, really highlighting, and this is sort of old news now, but really highlighting how remarkably, as David Bertioli would say, how remarkably similar peanut varieties are to each other. And so this is another example highlighting that. And what we're really interested in is sort of the variation and mapping the variation. So what we did was we used the PacBio long reads and comparative genomics with the assembly to look for structural variation. And so it was really interesting in that there was a large number of large insertions and large deletions. So 719 large insertions greater than a kilobase, and almost 1,600 large deletions. And the median size of the insertions was around 8,400 kb, and the median size of the deletions was about 41 kb, which is a lot of sequence. Also, many small indels, smaller than 1 kb, with median size 30 and 22. So these structural variants, and I'm more interested in the large ones because they can contain whole gene sequences and multiple genes. These really are representing a large amount of variation relative to what we usually think about in terms of variation, which is SNPs. And again, when you look at the comparison between NC94022 and Tifrunner, it is remarkably similar. Only one SNP per 10 kilobase of sequence between them. Could only find 275,000 SNPs in the entire genome. So that is just an astonishing result. And so, what does that tell you when you're trying to do fine mapping after QTL mapping? What it tells you is that if you have a parental genome sequence between your resistant, if that's what you're looking at, and susceptible varieties, there's a very small amount of variation in that region, even if it's very large that you identified. So that unlocks this kind of amazing potential to go straight to functional variation, where in other crops it's much harder to do because there's so much more, so many more differences between the two in those regions. So if we look at the FineMap region in this case for TSWV, it's 250 kb, which includes 26 predicted gene models. So close to 26 genes in this region, 250 kb, lots of potential for differences, but there's only 8 SNPs in the entire region. 8 SNPs and only one large insertion. And it just so happens this large insertion is incredibly interesting. So there's 6 predicted genes in this insertion, 2 genes that are duplicated. So 2 genes that are next to each other that is then duplicated, so a duplicated cassette. One of the genes is a kinetochore protein, and the other gene is a glutamate receptor like 3, which I'll talk about on the next slide. And what we did was we generated an indel marker that selects for this insertion. So I'm showing over here, it's 3 primers where there's a common primer and then a specific primer for the NC94022 within the insertion, and then for Tifrunner or sonoleic, a piece of DNA that is on the other side of the insertion. And unfortunately, that region is very similar, and in NC94022, it has that particular sequence downstream, and so when you use these primers, you get the insertion primer that is generated, but also the common primer as well. And so this is a really robust way to look at which individuals. Actually, my son wants some milk, if you can't hear. Individuals have this particular insertion. So what's interesting about this? So this glutamate receptor like gene actually is found in a Nature article that came out this year. Actually, it mediates systemic acquired resistance, so leaf-to-leaf signaling after wounding, and so this is an incredibly strong candidate variation for providing resistance to TSWV, which, as you know, comes into the plant through the Thrips vector. So the lessons learned here, like I said, because of the remarkable similarity between any two peanut genotypes, having that de novo genome sequence is incredibly powerful, and being able to generate it now is really much simpler than identifying SNPs with short reads or running SSRs. This is something that everybody can do at a relatively low cost. Again, showing the cultivated peanut has very little energetic SNP diversity, the same exact estimate for SNPs between the 2 cultivars as we got with shiitake that was published in Matters Arising in Nature Genetics, showing very clearly the genetic bottleneck that happened in cultivated peanut, and that cultivated peanut is a very young species. And again, having that genome really greatly increased the probability of getting that functional variation, and getting the gene and getting a marker that unlocks the potential of marker-assisted selection. So it's a really exciting result. And most of this funding, I believe, is from USDA ARS, from Baoju, as well as sequencing and Brian Scheffler's group. Peggy and Corley are providing a lot of the phenotyping data, and so I wanted to acknowledge all of their groups, and thank you very much.